In this lesson, we'll discuss osmosis and tonicity, and here are your objectives. First, to define osmosis. Osmosis is the movement of water across a semi-permeable membrane, meaning a membrane that lets some things through but doesn't let other things through. And you'll sometimes see that written as selectively permeable as well. They're often used interchangeably. Now, in order for osmosis to occur, the membrane must be impermeable to some solute, some given solute, meaning it doesn't let it through, but it must be permeable to water, meaning it lets water through, and we'll see why that is the case in order for osmosis to happen. Now, osmosis is often introduced with U diagrams like are seen here. So imagine that we had a semi-permeable membrane right there, and we had water on both sides, but in the water on this left side, we had more particles of a solute than we did on the right side. So this is all water, aqueous solution, but the aqueous solution is more concentrated on the left side. And importantly, this solute cannot get through that membrane. The membrane is impermeable to that solute. Now, we have a solute in higher concentration on the left and lower concentration on the right. And what's going to happen here, in an effort to equalize those concentrations, is because the solute itself can't move, water is going to move instead to try and make the concentrations equal. So we will end up with something like this, where no solutes have changed sides, but water has moved. In this case, water has moved this way to try and equalize the concentration. And you can see, as I've illustrated here, that we end up with more water on the left side. And I've tried to show that the concentrations of solutes are equal. So the water can't move, the solute can't move, so the water moves instead in an effort to try and equalize the concentrations. Now, important to note that if the solute could have moved, that's just what would have happened. We wouldn't have seen movement of water. But because the solute couldn't cross the membrane, water moved instead. So what we're seeing is that water is moving from an area of higher water concentration. In this case, the right-hand side in the diagram is an area of higher water concentration. That's similarly an area of lower solute concentration, to the left side, which is an area of lower water concentration, or higher solute concentration. So it's still following the same kinds of rules as we saw for diffusion, but now we're considering water. The solute can't move because the membrane is impermeable to it, so the water moves instead. By the way, you'll also often see this written as higher free water concentration and lower free water concentration. And that's because if you look at a molecular level, what's happening around each of these solutes is you're getting water that is forming a solvation shell around them. And that water is basically being taken out of consideration for moving across the membrane. Because more of the water molecules here on the left side are taken out of consideration because they are busy being in this shell around the solute molecules, they're less free to move from the left to the right. And at a molecular level, that's why we actually see movement from the right to the left, from higher water to lower water concentration, because there's more water available that's not in these solvation shells, in these shells around the solutes on the right-hand side. So that's where the word free comes from. Free means the water that is not around the solute. So we can see then that when we're figuring out whether osmosis will happen, both solute concentration and membrane permeability matter. It's not just about solute concentration, because if we had a high concentration on one side and a low concentration on the other of a solute that could freely pass through the membrane, then no osmosis would happen at all. It's only when we have solutes that cannot pass through the membrane that we end up with osmosis. Just a quick note that in chemistry you will have come across measuring solute concentration in moles per liter, and in biology, when we're thinking about osmosis, we often measure in osmoles per liter, and the number of osmoles is the number of total particles in solution. As a quick example, if you had 2 moles of sodium chloride in 1 liter of solution, you would say you had 2 molar sodium chloride, 2 moles per liter. That's what the big M means. Now, if you had 0.2 moles of sodium chloride per liter of solution, in other words, 200 millimoles of sodium chloride in solution, you'd say you had 200 millimolar sodium chloride. Again, 200 thousandths of a mole, 200 millimoles in every liter of solution. But because sodium chloride breaks into two particles, sodium ions and chloride ions, and because that's what we really want to know to determine which way the water is going to move, we need to count up the total number of particles like we see here. We calculate osmolarity by counting up the total number of particles, and we have 200 milliosmolar sodium and 200 milliosmolar chloride, so in total we have 400 milliosmolar sodium chloride in this same solution. These are different ways of expressing the same thing depending on what we're interested in. If we're interested in figuring out whether and how osmosis is going to occur, we need to think about osmoles. Now, whereas molarity and osmolarity give us numbers, they're quantitative, the important concept of tonicity is qualitative. So tonicity is a way of describing a solution relative to a cell that we put in it. So we're always describing a solution. And it's really just telling us which way water is going to move. And therefore, we're categorizing a solution into one of three categories: hypotonic, isotonic, or hypertonic. And again, these are categories of which way water is going to move. Let's put a cell in each of these beakers. Now, the only solutes I'm going to draw are going to be solutes that cannot cross the cell membrane. These are called non-penetrating solutes because they cannot get through the cell membrane. In an isotonic solution, we have equal concentrations of these solutes both inside and outside the cell. And therefore, there is equal movement of water out and into the cell. And so we have no net movement of water. There is movement of water across the membrane, but there's no net movement of water, and so the cell is not going to shrink or grow. In contrast, in a hypotonic solution, we have more of these non-penetrating solutes inside the cell than we do outside. And so as a result, in order to try and equalize the concentration, water is going to move in by osmosis because the solute can't move. We have, in a hypotonic solution, a net movement of water moving into the cell. In contrast, in a hypertonic solution, we have a higher concentration of these non-penetrating solutes outside of the cell, and again, because they can't move into the cell to equalize the concentration, water is going to move out, like so. So, in a hypertonic solution, water in net moves out. Water moves in hypotonic, there's no net movement of water in an isotonic solution, and water moves out in a hypertonic solution. The consequences for the cell in a hypotonic solution over here, if water is moving in, let's say the cell has no cell wall like an animal cell, if enough water moves in, then the cell is actually just going to burst, and we call that lysis. If it were a plant cell or a bacterial cell or a fungal cell that has a cell wall, that cell wall provides structural rigidity, and instead the contents, the cytoplasm, just push up against the cell wall, forming a rigid structure, and we say that the cell is turgid. That's like celery that you've placed in water. Water is a hypotonic solution to the cytoplasm of the celery. The celery gets more rigid, and the word for that is turgid. In contrast, in a hypertonic solution, when water is leaving, if there's no cell wall, the cell is just going to shrivel, like in an animal cell. That's what you'd see. In a cell with a cell wall, like a plant cell, fungal cell, or bacterial cell, instead you're going to see the cytoplasm shrivel, but the cell wall itself will maintain some of its structure. and that's visible under a light microscope, or can be, and that's called plasmolysis, that shriveling of the cell membrane and the cytoplasm away from the cell wall. So that's an introduction to osmosis, osmolarity, and tonicity.